What would you do to prevent a personality disorder that causes the lack of remorse, the tendency to manipulate, and callous disregard to others? Hello, I'm Sarah, and this is a literature review on the causes of antisocial personality disorder. Antisocial personality disorder, also called ASPD or sociopathy, causes the lack of empathy, aggression, and the excessive manipulation of others. Such symptoms expose the society to violence and harm. Individuals with ASPD often face the consequences of imprisonment, difficult relationships, homicidal and suicidal behavior, and early death, typically due to violence. Unfortunately, the cure is still unknown, and the severe effects of ASPD put everyone at risk, including you. Thus, it's imperative to investigate its root causes, or etiology. This literature review will demonstrate that there are various genetic, epigenetic, and environmental causes of ASPD. For background, genome-wide association studies, or GWAS, are types of studies that identify a certain genetic variation that could be linked to a trait, in this case, ASPD. These specific variations are called single nucleotide polymorphisms, or SNPs. One GWAS investigated which SNPs could lead to ASPD by comparing the DNA of 370 criminals with ASPD and over 5,000 healthy participants. They noticed that this key SNP, among many others, was genome-wide significant, meaning that this variation was found in people with ASPD only. This indicates that it could be involved with the disorder. This SNP was intergenic of these listed genes, which are expressed in the frontal cortex of the brain. This plays a role in fear conditioning, stress response, and decision-making. If this SNP alters these genes and interferes with these mechanisms, it may lead to the poor development of conscience, antisocial responses to the understimulation of fear, and risky behaviors, all of which are symptoms of ASPD. As shown, several genetic variations can increase the risk of ASPD. While genetic variations may contribute to ASPD, environmental risk factors also play a role in its development. A study revealed that too much TV viewing during the childhood dramatically increased the chances of getting ASPD. Young adults had significantly higher chances of criminality and ASPD diagnosis when they spent most of their time watching TV in their childhood. They theorized that this is due to the observational learning theory, in which humans imitate and internalize actions seen on the television. When people are exposed to violent content, it leads to desensitization towards brutality, aggressive behavior, and ASPD symptoms. Another risk factor depends on the quality of parental or maternal care in one's early life. If a mother or parent fails to verbally interact, personally soothe, and greet an infant, this could lead to disorganized attachment and antisocial behavior later on. Finally, psychosocial deprivation, such as the lack of socialization, neglect, and abuse, increases the risk of developing ASPD. Psychosocial deprivation decreases the size of the prefrontal cortex and thalamus. A smaller prefrontal cortex leads to trouble with behavioral control, inhibition, and reasonable decision-making, while a deficit in the thalamus volume leads to difficulty in extinguishing thoughts about past trauma. Overall, television, maternal withdrawal, and psychosocial deprivation are environmental risks for developing ASPD. Now that we know the environmental risks, how exactly do they lead to ASPD? How do they cause mechanisms that cause ASPD? For background, the epigenome is a collection of chemical tags, in this case methyl, that edit gene expressions without actually changing the DNA. These chemical tags attach into the DNA, which is the process of methylation. Methylation blocks transcription and gene expression, causing certain functions to be interrupted. Environmental stimuli, such as social interactions, stress, and the previously mentioned risk factors, influence methylation. Thus, the epigenome adapts in response to environmental stimuli, which can result in the development of ASPD. The following study compared the epigenomes of 86 male offenders with ASPD and 74 healthy controls. The researchers discovered that methylation of the MAOA gene was strongly associated with ASPD. That gene regulates the production of monoamine oxidase A, which is an enzyme that breaks down monoamine neurotransmitters, such as serotonin. They discovered that insufficient gene activity led to serotonergic system dysfunction and antisocial personality as a result. They also found a region of interest in the DNA of criminals, and hypermethylation of this region blocked transcription and gene expression. This decreased the production of the enzyme and induced abnormal serotonin levels, which finally led to antisocial behavior. As shown, the root cause of this sequence is the environmental stimuli, since the epigenome adapts to it. Prevention of ASPD from epigenetics depends on the exposure to these certain risk factors. Overall, hypermethylation on the MAOA gene due to the previously mentioned environmental risk factors amplify the risk of developing ASPD. With this knowledge of the genetic, environmental, and epigenetic factors of ASPD, how can we prevent this disorder from spreading in the society? Primarily, individuals who have genetically inherited a SNP, leading to antisocial behavior, may keep their behavior under control by taking medication or therapy. Additionally, limiting television viewing, exposure to violence, and adverse experiences in children may prevent the development of the disorder due to environmental factors. Finally, demolishing environmental risks as much as possible may prevent the epigenome from causing antisocial behavior. While there's still no effective cure, applying our knowledge of the etiology of ASPD can prevent it from passing down to future generations. The various genetic, environmental, and epigenetic causes of ASPD open countless solutions to prevent it as well. Thank you for investigating these types of causes of ASPD with me. With this awareness, the actions we take may reduce ASPD in our society, and future research may discover an effective cure for it. Once again, thank you for listening.
Hi, my name is Rishika Kulkarni, and I'm a current junior in high school at Francis Parker School. Today, I will be talking about the review paper I wrote discussing the effects of music therapy on neurological disorders. So, what exactly is music therapy? Music therapy is defined as utilizing different components of music as a clinical treatment method. Unfortunately, it has not always been a popular concept. In the 1940s, Everett Thayer Gaston, now known as the father of music therapy, researched the benefits of music therapy on mental health. In 1968, he concluded that music therapy follows the path of a behavioral science. Additionally, he claimed that a music therapist's main goal is to develop an interpersonal relationship with the patient in order to observe the patterns in behavior change. Music therapy is found to affect three main sections of the brain: the cerebellum, frontal regions, and the hippocampus. And in this presentation, I will be going in depth of each of these three different sections. So first up, we have the cerebellum. If you look at the diagram on the slide, you can see that the cerebellum is quite small compared to the cerebrum and the rest of the brain. It is this tiny little purple region that the diagram is pointing to. So the cerebellum is frequently known as the little brain because of its small size compared to the cerebrum. It serves as the central processor for motor functions, therefore affecting movement. To ensure coordination and the accuracy of movements, the cerebellum receives information from the spinal cord and other sensory regions. Next, the sensory information is processed, which then translates to controlling motor functions. Additionally, the auditory system is closely connected to the cerebellum. Therefore, it also helps control movement, particularly with automatic responses. However, if the cerebellum were to be damaged, then the loss of functions would include balance and posture. So as you can see on this flow chart, you have the path that it takes to process information. And it is very clear that the cerebellum works closely with the auditory system to process information and therefore control these motor functions like movement. So how does music therapy affect the cerebellum? Neuroscientists have found that music can morph the brain, especially within the motor and auditory regions, by either forming new pathways within the brain or reinforcing pre-existing pathways, and these all contribute to heal the brain. Listening or even creating music activates motor regions such as the cerebellum and results in functional movement. Therefore, music can also be tailored to a particular individual, resulting in the activation of specific neural pathways. Particularly, the cerebellum controls timing of motor functions, particularly processing rhythm. This indicates that music can be altered based on the rhythmic nature and are then trained to control fine motor functions, which then increases movement within patients who experience a loss of control over motor functions. This is particularly very important, especially with different neurological disorders that affect movement, which I will be getting to in the next slide. So to test this hypothesis, a case study was conducted on patients with Parkinson's disease. Neuroscientists want to test if a patient's control over motor functions would increase if rhythm and timing were altered in music. Parkinson patients were asked to synchronize their speed of walking to rhythmic cues. As soon as patients coordinated their movement with these rhythmic cues, they were able to not only walk faster, but exhibit more control over their bodies. Their neuromuscular activation, limb coordination, ankle extensions were more consistent as well. Not only did altering rhythm increase coordination in Parkinson's patients, but it also maintained consistency in their movement. Furthermore, this improvement was far more drastic than anything that physical therapy could have done. So this would lead to the conclusion that changing components such as rhythm and timing to improve motor functions are far more better than physical therapy. And this is not only just in Parkinson's disease patients, but other patients who struggle with control over movement or are looking to strengthen their control over movement. Because as we said, the music can not only grow neural pathways, but it can also reinforce these pathways. Next, the frontal region, the second part that the music can affect. So the frontal region is not particularly shown in this diagram, but you can see this pink region, which is the prefrontal cortex, which I will be talking about more specifically as we get on with the presentation. The frontal region will cover a vast amount of structures, including the frontal lobe and the prefrontal cortex. So the frontal region, as the name indicates, is located at the front of the brain. It serves to control higher level functions such as attention, planning, memory formation, and most importantly, speech. All of these are known as cognitive functions. A particular cortex, known as the prefrontal cortex, is located in this frontal region. The prefrontal cortex plays a major role in decision-making in any executive functions. To do this, it initiates proper behavioral responses in order to interact with the environment surrounding it by evaluating different sources of information. So we know that the brain is able to receive several different sources of information from its outside environment, and what it does is that it's filtering these responses to send signals and then therefore control higher level functions. By doing this, the prefrontal cortex is able to control decisions when faced with challenging streams of information. Furthermore, it sends signals to other regions of the brain to assist the prefrontal cortex in making executive decisions. However, the prefrontal cortex takes a little bit longer to develop than other structures of the brain, which also contributes to more impulsive decisions made at a younger age. This also relates to why younger people are more prone to getting more influenced by their outside environment, just because their prefrontal cortex is less mature and takes longer to develop. Additionally, since it plays a vital role in memory creation, it's one of the first areas to actually be affected by degenerative diseases such as dementia and Alzheimer's, which I'll be doing a little case study on. So the frontal lobe and how it affects music therapy. Patients who have dementia can recognize songs that hold some significance to them. This is because certain regions of the prefrontal cortex that monitor musical endeavors shut down. However, regions that trigger initiation increase their activity. And so if you see on the right this image with the effects of music on prefrontal cortex based on gender, you can see how that you can show improvement with the baseline 10 minutes, 20 minutes, and 30 minutes, and how these bars are actually representing the standard deviation in these plots, showing that there's actually a pretty high standard deviation. So there are two different claims that scientists are using to strengthen their hypothesis that music therapy increases cognitive functions. So they were trying to figure out specific music exercises that would impact cognitive functions, such as speech through neuroplasticity. However, this connection is clearly not as obvious as the effect of rhythm on motor functions. Scientists began with two key ideas to explore this concept further. So the first is that the sharing of functions and the development of the auditory system, also known as the auditory scaffolding hypothesis, shown on the slide. The first insight states that the brain's underlying music is used to share with other functions of the brain. This would mean that the music stimulates speech and other cognitive functions regions differently than other systems. There are two ways music can stimulate the brain: bilaterally, which means equally, or on the right hemisphere more than the left. For example, in injuries that occur solely on one hemisphere of the brain, music is able to form more flexible neural pathways to either strengthen or relearn these functions. The other idea is to use the auditory scaffolding hypothesis. This states that the functions that include timing and processing are sent to the auditory system, which processes time-sensitive information. Cognitive functions require processing time-sensitive information and complex temporal organization. This is why scientists believe that listening to music can provide a scaffolding, basically, for either enhancing or relearning certain high-level functions, such as speech or any other cognitive functions. Since music is actually considered a complex temporal auditory language, it can actually enhance these cognitive learning by using the auditory scaffolding model. So now to the case study on memory. In a memory study that was conducted by scientists, word lists were actually embedded in songs, and they were shown to activate the frontal region as well as the temporal lobe, in contrast to only word activities without music. So oftentimes we have conventional therapy, which just uses verbal expression without any type of music or song. So now scientists are trying to explore how, if we combine these written therapies or more verbal therapy with music therapy, how it affects both the frontal region and temporal region. So this is activating only the left hemisphere when you are using just verbal therapy. So this shows that music therapy involving elements of spoken word can actually enhance several regions of the brain instead of just one, compared to conventional therapy. Moreover, music can also stimulate both neural networks on both hemispheres of the brain, which was shown to actually increase attention span, which is often decreased in patients who have experienced traumatic brain injuries, such as a stroke. After using this reasoning and conducting a study, researchers found that therapeutic music exercises indeed do improve speech in patients who have had traumatic brain injuries, strokes, and aphasia. Next, we have the limbic system. So this is the final part of the review paper that I will be discussing. So as you can see on this diagram, the limbic system is also not explicitly stated, but I will be getting into two key components of it, which is the hypothalamus and the amygdala, which is shown in both the diagrams. As you can see, the amygdala is this orange area right here, and the hypothalamus is this green tiny area that it's pointing to. So the final but developing aspect of neurology that is affected by music therapy is mood. So the hippocampus and a structure known as the amygdala are a part of the whole limbic system, which functions to process memories and manage emotional reactions. Specifically, music triggers emotional responses that are rapidly processed by various neural networks. If an emotion is correlated with a past memory, for example, the neural networks that are activated will be different from a spontaneous response. The hippocampus serves as a huge role in memory processing, specifically in sorting short-term memories from long-term memories. Since its main function is to process memories, any damage to the hippocampus will result in a loss of memory, leading to degenerative memory disorders. Just as the limbic system is associated with emotions and past experiences, the hippocampus also relies on life experiences to connect with music. So as you can see in this flow chart, we can see that emotional responses can either be based off of past memory, or it can also stimulate a new response. So with the amygdala, it controls response time to events. For example, we all know about the fight or flight response. It's often a term that's loosely tossed around, but what exactly is it? So the amygdala is able to send a signal to the hypothalamus, which then activates the sympathetic nervous system, a system of nerves that work in collaboration with the amygdala to carry out this fight or flight response. Since the amygdala is responsible for initiating spontaneous responses, it also connects memories with emotional encounters. So for example, if the amygdala were to experience any sort of damage, an individual would not be able to recognize emotion in music. When contrasting the emotion of music, so sad or happy, the amygdala has an instantaneous reaction to the mood of music, perceiving it as either pleasant or depressing. This happens through the release of dopamine, a neurotransmitter that triggers feelings of joy. Because of this, an individual's serotonin levels will also increase, thus will decrease the negative emotions you experience. By focusing on melody and mood of the music, the limbic system is activated, stimulating several neural networks in the brain. So, on a case study on autism patients, an occupational therapist conducted a study with a group of children attending a specialized school for several disabilities, one of them being autism. They attended a music therapy class 3 times a week, and they were clearly excited even before attending the class. This is a particularly important detail to note as we progress through the study. In class, they experimented with singing songs, expressing emotions, and playing with several elements of music. After observing the children for a while, the therapist concluded that the students who had autism once struggled with interaction were now more social with their peers and teachers. Additionally, this motivated them to participate more, not only in school, but also at home. This led to therapists believing that music therapy improves social skills and emotional intelligence in children with autism, especially by altering these elements of music such as mood and melody. So now to conclude this presentation, what can we take from this? First, the results. This review paper determined that music therapy does have neurological benefits on movement, cognitive functions, and mood by analyzing specific regions of the brain and their functions. The cerebellum, frontal region, and limbic system all play a crucial role in reaction to music, and they're certainly not the only structures that are impacted by music. Secondly, it was determined that specific qualities of music were required to be changed, such as rhythm for movement, timing for cognition, and melody for mood. This is especially supported by the 3 case studies which experimented on patients with Parkinson's, memory loss, and autism. And lastly, the 3 conclusions that were determined above can be weaved together to determine that music therapy is indeed a better alternative to conventional therapy, producing drastic improvement in patients. So further study, there is still a lot more research about the brain and is an ever-changing nature. Music therapy is just one part of the puzzle, with possibilities ranging from any sector of art therapy. If music therapy works, then who's to say that other forms of art therapy can't work? As the nature of therapy changes, it's important to understand that mood and emotion are still an unexplored area. While neuroscientists can do their best in predicting an emotional response, we still don't know how consistent these responses are or if they're constantly changing. Furthermore, while the argument made in the emotions section of this presentation heavily relies on music affecting an individual's life experience, we don't exactly know the correlation of the limbic system and how it processes or sorts previous occurrences. Lastly, scientists are currently wondering if music therapy can heal an individual's brain. It is technically inducing a mood or emotion. This is quite an area that is unknown for us, and we don't know the limits that inducing false emotions has. Doing further research on all these topics because it could eventually break the barriers surrounding conventional therapy, introducing more alternatives to our society. And some action items. For some people in the audience, the neurological aspect of this presentation was absolutely boring. Here are some things you, the audience, can do to support music therapy. First, do your best to support any organization surrounding music therapy or art therapy. Whether that be donating or spreading awareness, everything you do will definitely count as a step into the future of therapy. Social media is one of the greatest platforms to spread awareness about the neurological benefits of music therapy. Lastly, I'd like to say I'm the founder of a nonprofit organization called Art for You based in San Diego, California. Volunteers and I work to host drum circles for kids, performances for the elderly, and art classes for all ages to induce therapeutic mechanisms of expression using art. Our website is on the slide, and our Instagram page is also on the slide. Do your best to spread awareness, and even feel free to join. We accept all volunteers. And now for my works cited, it's quite lengthy, so here's page 1, page 2, and page 3, and one more page. And finally, thank you. I would like to especially thank Polygens and my mentor Adrian for helping me and putting so much time into meeting with me and helping me prepare this presentation. Thank you so much.
Yeah.
Hi, my name is Rishika McCarthy.
Hi, my name is Niki Itharaja.
Hi, my name is Itharaja.
And this is our presentation on lupus, understanding the pathophysiology of underfunded autoimmune diseases. So the immune system. How does the immune system work? Well, the immune system is made up of cells, organs, and proteins that work together to defend the body against foreign invaders, such as viruses, bacteria, and fungi. There's the innate, passive, and acquired immune system. In general, when the body senses foreign substances called antigens, the immune system works to recognize the antigens and tries to get rid of them. B lymphocytes are triggered to make antibodies, also known as immunoglobulins, and these proteins lock onto specific antigens. After they're made, these antibodies usually stay in our bodies in case we have to fight the same germ again. That's why when someone gets sick, they usually won't get sick from that same disease again, because you're immune to it and you still have those antibodies in your body. Although antibodies can recognize an antigen and lock onto it, they can't destroy it without help. And that's where the T cells come in. Also known as killer cells, they destroy antigens tagged by antibodies or cells that are infected or somehow changed. And these T cells can also help signal other cells, such as phagocytes, to do their job.
However, there are times when the immune system attacks and kills its own healthy tissues and cells, a condition known as autoimmunity. An autoimmune disease occurs when the immune system doesn't know the difference between the body's own cells and tissue and foreign pathogens. For example, the image on the slide shows the leftmost kidney inflamed with lupus, an autoimmune disease, while the one on the right is completely normal.
Lupus. Lupus is one of the hardest diseases to diagnose because of the complexity of the disease and how its signs and symptoms overlap with other disorders. Therefore, multiple tests have to be taken by a patient to be fully diagnosed with lupus. The combination of blood tests and urine tests with signs and symptoms and a physical diagnosis can help find lupus in someone's system. An examination of a sample of your urine may show an increased protein level or red blood cells in the urine, which may occur if lupus has affected your kidneys. Diagnosing lupus is not easy as signs and symptoms vary from person to person. There are many symptoms of lupus, but some major ones include fever, fatigue, and skin issues. One major skin issue is a butterfly-shaped rash across the cheeks and the bridge of the nose, as well as rashes throughout the body. Another skin issue is skin lesions that appear or worsen with sun exposure. There can also be pain in joints, muscles, chest, while breathing, and stiff joints.
So in regards to treatments and medication, there has been a significant amount of studies and trials done in regard to cures and more efficient management of lupus because there is no official cure for lupus. So there was a clinical trial that was started in 2017, and it studied the effectiveness and tolerability of JBT-101, or lenabasum tolerance, in individuals with systemic lupus erythematosus. A research study was conducted on the pipeline to automate classification of glomeruli features in renal biopsy, which basically is a biopsy of a part of a kidney. As for the results, the researchers proposed a complete pipeline for the classification of WSIs into various LN stages based on those observed glomeruli features. There's also a pilot study of CC220 to treat systemic lupus erythematosus, and results indicated that the drug had improvements to be made, but it was more successful than the placebo. A possible route to creating treatments for those with lupus nephritis could involve researchers further studying the genetic cause and drivers of the expansion of the Bowman's capsule. This is important because the expansion of the Bowman's capsule leads to protein leakage in the glomerulus, which is evidenced by how many patients' urine tests have extremely high levels of protein in them.
Some side effects of medication include stomach issues or digestion problems, such as diarrhea, nausea, or vomiting. Other side effects of treatment also include a decrease in the number of blood cells, fever, bleeding, and increased susceptibility to infections and other medical complications. These side effects should go away as the body adjusts to the medication. In fact, having such side effects are a positive sign, as it shows that the medication is actually working on the body.
Funding and awareness. So another important part of this presentation is talking about funding. So the two main funders for lupus are the Lupus Foundation of America, which secures lupus research funding through the Department of Defense's Congressionally Directed Medical Research Program. Both of these funders get money from the National Institutes of Health. Lupus gets about $139 million in funding as of 2022. While lupus gets around $130 million in funding, cancer gets about $6.25 billion of funding from the National Cancer Institute. $139 million funding to dig deeper into lupus is a fair amount of money compared to the $90 million that was given in 2014 as observed in the graph. Although cancer is a more common disease and therefore receives much more funding, lupus still needs more money in order to recruit high-achieving and credible researchers who can deliver the most accurate information on this chronic disease.
So the next steps to take are raising awareness about autoimmune diseases such as lupus and other autoimmune diseases such as multiple sclerosis, so that we can raise more research and funding and find permanent cures and better treatments for people with autoimmunity.
Autoimmune diseases such as lupus are underfunded and need more attention. Although other diseases such as cancer are more common, autoimmune diseases are still a major problem. According to the American Autoimmune Related Diseases Association, autoimmunity is a poorly understood category of health that is comparable to heart disease and cancer. Annually, over 50 million Americans are affected by an autoimmune disease, and patients spend around $100 billion on autoimmunity. The number of autoimmune cases and the cost of treating them will only rise in the future. By raising awareness and funding for autoimmune diseases, it can be easier to find future solutions to autoimmune diseases such as lupus. These are our references, and we would like to acknowledge Dublin High School, Polygents, and our mentor, Gabriel Rodriguez. Thank you.
Hi everyone, my name is Akanksha, and today I will be presenting my research on the CRISPR-Cas9 gene editing system through an exploration of CRISPR-Cas9, a literature review of the gene editing system's applications in treating diseases in humans, and an experimental lab report of its use in transforming E. coli bacteria. This presentation encompasses two projects that I did with Polygents. One is a literature review article containing research on CRISPR-Cas9 disease treatment, and the other is an experiment that I did recently myself with CRISPR-Cas9 transformation. As shown by the timeline above, CRISPR technology has been in development for a while, but it is only recently that scientists have discovered how much of an incredibly useful gene editing tool CRISPR-Cas9 truly is. Based on its performance in clinical and preclinical studies focusing on disease treatment, it is applicable in everything from cell line knockouts to overall health improvements to the elimination of entire genes to treat diseases. Once potential accuracy, specificity, and efficiency issues are resolved, CRISPR-Cas9 will greatly benefit the biotechnology and health industries. Overall, the significant genomic advantages that CRISPR-Cas9 has provided to the scientific community truly make the gene editing system a technology of the future. I will first be presenting my literature review, which focuses on CRISPR-Cas9 background information, current and past clinical research on the applications of CRISPR-Cas9 as a disease treatment, scientific and social considerations of CRISPR-Cas9 that must be addressed, as well as future directions of CRISPR-Cas9. So what is CRISPR-Cas9? CRISPR itself stands for clustered regularly interspaced short palindromic repeats, and CRISPR-Cas9 is a type of gene editing system discovered in the early 2010s. It originates from a bacterial immune defense system against viruses that involves the Cas9 endonuclease enzyme, single guide RNAs or sgRNAs, and PAM recognition or protospacer adjacent motif recognition. So out of the three known types of CRISPR systems, CRISPR-Cas9 is a type 2 system from the Streptococcus pyogenes or S. pyogenes bacteria. In the CRISPR-Cas9 system, an sgRNA-based dual structure binds with the Cas9 enzyme, the preferred genome editing enzyme, to form a ribonuclear protein which binds to the DNA at its target. Once a PAM sequence is recognized and RNA and DNA structures called R loops are formed, DNA cleavage and genome editing are initiated. So in my research paper, I studied the clinical and preclinical applications of the CRISPR-Cas9 gene editing system in disease treatment. The above figure shows the specific diseases that I researched, which included various phases of clinical CRISPR-Cas9 trials with sickle cell disease or SCD, TDT, and lung cancer, as well as preclinical research with HIV, Duchenne muscular dystrophy, Huntington's disease, cataracts in mice, and the coronavirus in hamsters. The results and outcomes of each clinical trial with CRISPR-Cas9 proved to be very positive overall. In both SCD trials, it was found that the CRISPR treatment reduced SCD symptoms and complications. In the TDT trial, complications of TDT were resolved as well. In the lung cancer study, there were only lower grade symptoms occurred in the study participants and no major ones, and they also had a greater survival rate as well. Thus, CRISPR-Cas9 mediated gene therapy appears to be a successful strategy for treating SCD, TDT, and lung cancer, and it may be an effective disease treatment in the future. With positive clinical and preclinical outcomes, CRISPR-Cas9 appears to be an effective disease treatment, but certain issues with CRISPR-Cas9's efficiency, accuracy, and specificity must be resolved before it can be made publicly available. The sgRNA, the PAM sequence, and the Cas9 enzyme must all be effective in order for the process of CRISPR-based gene editing to occur properly. Another issue with CRISPR is its safety, as current safe clinical trials have been limited, and various CRISPR-Cas9 therapy techniques have come under fire due to a concerning amount of off-target effects. Furthermore, with the idea of designer babies and CRISPR-Cas9's cosmetic potential, the gene editing system has often been deemed highly unethical. There have been significant efforts and progress made to overcome these scientific and moral concerns, however. For one, various validation techniques have been built to minimize off-target effects, and CRISPR-Cas9 bacterial immune systems themselves have actually evolved these self-contained accuracy checkpoints. Additionally, scientists have created sgRNA libraries and predictive algorithms as a way to predict off-target effects with CRISPR systems. As for the ethical issues, it is clear that a boundary must be established between somatic disease therapy and cosmetic germline editing in order for the scientific community to continue developing CRISPR-Cas9 gene editing system. Overall, once CRISPR is deemed safe and effective in using in disease treatment, it will revolutionize the way medicine and the health industry works. So after I researched the use of CRISPR-Cas9 in disease treatment, I wanted to see it hands-on for myself. So I conducted an experiment using CRISPR to genetically edit and transform bacteria to become antibiotic resistant, so it could actually grow on an antibiotic containing plate. I wrote a lab report on this experiment, which is pictured above, and I will be presenting it now. So in this experiment, I successfully used the CRISPR-Cas9 gene editing system to genetically modify the 43rd amino acid in the RPSL amino acid sequence of non-pathogenic DH5 alpha E. coli bacteria. This genetic mutation allowed the bacteria to become antibiotic resistant and survive on LB media containing the antibiotic streptomycin, which normally prevents bacteria from growing on it. Overall, the lab experiment took 3 days, as pictured in the timeline above, including a preparation day before and a results measuring / cleanup day after. The first experiment day was dedicated to making and pouring these LB agar plates, some without antibiotics and some with, where the bacteria would be plated and grown. Towards the end of the first day, I plated untransformed bacteria on an LB plate and let it grow for about 18 hours. The second day of the experiment focused on transforming the grown bacteria by adding it and other CRISPR components to a bacterial transformation buffer and incubating the solution for approximately 12 hours. On the third day of the experiment, I spread the bacterial solution around on an antibiotic LB plate and left it outside for about 84 hours, waiting for the transformed bacteria to grow. The above images show the progression of growth of these medium-sized yellow circular colonies of the transformed bacteria on the antibiotic plates after approximately 84 hours of incubation. There was almost no growth for the first 18 hours of incubation, as you can see, but smaller spaced out white dots began to appear on the plate shortly after the initial incubation period. And the dots became larger and more yellow and grew faster over time. Ultimately, this means that the experiment was successful and the CRISPR-Cas9 gene editing system genetically mutated and properly transformed the E. coli bacteria, allowing it to grow on an LB plate containing streptomycin. Beyond the bacterial transformation experiment that I did, I would like to use the CRISPR-Cas9 system to investigate the knockout of the muscle growth inhibiting myostatin or MSTN gene using a similar experimental procedure to the E. coli transformation. This experiment would require me to A, design a functional sgRNA sequence for the target DNA, as well as B, measure the impact of knocking out the myostatin gene in human cell lines or an organoid line. Inhibiting myostatin has shown to have the potential to treat muscle atrophy, improve disease resistance, and increase muscle growth so far. It has never been done in humans, so this would be a novel experiment and a significant step in CRISPR-Cas9 disease treatment. In conclusion, the CRISPR-Cas9 gene editing system is a very valuable tool in the scientific fields of study. Due to its success in clinical and preclinical studies, it may one day be used as a publicly available disease treatment pending further testing in cell lines and the resolution of certain safety and ethics issues as well, or such as off-target effects and cosmetic editing. It can also be used on a smaller scale to study bacteria and diseases themselves, to better understand the implications of antibiotic-resistant bacteria. In the end, the CRISPR-Cas9 system will greatly improve health and disease treatment in the future, and it will be a significant part of subsequent efforts to significantly better the human race overall. Thank you.
Hi, my name is Sophia, and I'm here to talk about the advantages of iPSC-NK cell therapy. Cancer is a leading cause of death. In 2020 alone, 20 million people died from various types of cancer. Part of what makes it so deadly is that it's composed of cells from our own body and are able to evade our own immune system. But new technologies make beating cancer possible, and one such is iPSC-NK cell therapy. iPSC stands for Induced Pluripotent Stem Cell. NK cell stands for Natural Killer Cell. It's a type of immune cell that is able to attack and kill different types of pathogens or cancer cells. How do NK cells work? NK cells are activated or inhibited through a series of receptor and ligand interactions. Activation is when receptors respond to specific ligands that are presented on pathogens or infected cells or cancerous cells. And when these receptors bind to these specific ligands, the NK cell receives an activation signal that prompts it to attack the target cell. Some examples of receptors include CD16 and NKG2D. However, inhibition works in a similar fashion. There are inhibitory receptors that respond to inhibitory ligands. These are known as HLA ligands, and they are present on every one of our cells. This means that when our normal cells are coming into contact with NK cells, there is no activation because the inhibitory receptor will see that there is an inhibitory ligand present and identify our cell as a normal cell, a healthy cell, which means that the NK cell will not attack our normal human body cells. In addition, cancer cells do not usually possess an HLA ligand, which means that the cancer cell can only activate an NK cell rather than inhibit it. This is part of a process called immunosurveillance, in which our white blood cells are able to travel through the bloodstream to go to various parts of our bodies to check for cancer cells. However, when the cancer cell mutates to a form that is able to evade the immune system, that's when tumors start to form. Tumor evasion can happen in 3 ways. First is when the cancer cell is mutating, there is HLA ligand preservation. Usually, and when cancer cells mutate, HLA genes are not preserved. However, in some mutations, this could be the case. Another way is the cancer cell will develop a type of non-classical HLA ligand, which the NK cell can still interpret as an inhibitory ligand. The third way is cancer cells can send out immunosuppressive factors such as TNF-beta or adenosine, which can suppress NK cell function. The point of cell therapy is to insert enhanced immune cells to aid the immune system against cancer cells. Some existing types of cell therapies include CAR T cell therapy, which is the use of T cells, another type of immune system, to battle cancer. Types of NK cell therapies include using NK cells from peripheral blood, umbilical cord blood, from the NK92 line, or iPSC. iPSC-induced pluripotent stem cell is a specialized cell that has been artificially programmed by several genetic factors back into a stem cell. These stem cells can then turn into whatever cell that the scientists want. With this technology, there is hope for something called an off-the-shelf therapy, meaning that there's a universal cell therapy that every patient can use. There are several advantages to the iPSC-NK cell therapies. The first of which is that there are endless sources. What does this mean? Other therapies, such as peripheral or umbilical cord blood, are autologous, meaning that only one person is able to use the cells that are developed from the process. These cells come from one person's own bodies, and if they're transferred to someone else, there could be a negative immune system reaction. So these autologous therapies are specific only to the donor. There's only one source of cells for one person. This is highly unavailable to patients without usable blood or don't have their umbilical cord blood. For example, patients on chemo or radiotherapy have very low percentages of NK cells to use that are present in their blood. So there's a need for universal cell therapy. iPSC can bring about this because they can be derived from any type of cell. For example, a skin cell, which duplicates frequently, can be used to create a stem cell that can then differentiate into an NK cell. These cells are allogenic and can help patients from all across the world because they're able to be used without harming the immune system in any way. There are unlimited resources because anyone's skin cell, can be used to create iPSCs for everyone to use. A second advantage of iPSC is the convenient lab use. iPSC has, first of all, self-renewal properties, which makes this easier for scientists to cultivate and duplicate NK cell populations. They also don't lose their cytotoxic properties, which usually happen when scientists try to cultivate normal types of NK cells. Second advantage is that they have a large homogeneous population, which makes genetic modification and experimentation much easier. And this makes it easy to make a large batch of the same product for a universal therapy. And finally, iPSCs are not as affected by cryopreservation, which preserves their cytotoxicity. The third advantage of iPSC-NK cell therapy are the numerous genetic modifications. These genetic modifications serve to enhance cell attack functions and overcome the tumor environment. For example, immunosuppressive factors or hypoxia, which is a lack of oxygen. Examples include attaching a CAR. CAR stands for chimeric antigen receptor, which helps NK cells locate the cancer cells more properly. Other ways include inhibiting genes that code for inhibition receptors on the NK cell, which then allow the NK cells to more easily be activated and attack cancer cells. So what exactly is the conclusion here? Although NK cell therapy, and especially iPSC NK cell therapy, is a relatively new field of study that scientists have to learn and study about right now, there is much potential for the future for iPSC and NK cell therapies to, one, be able to help more and more cancer patients achieve their dreams of battling the disease, and two, create a universal cell therapy that can be used by everyone and make battling cancer a more convenient thing to do. Thank you.